Welcome back to my YouTube channel. This is Samya. In this video, I'm going to solve the KSET license question paper from 2015, and this is the part 2 video, and I'm solving the paper 2. Now, without making any delay, let's begin with the video. Which of the following is a virus-induced cancer? Option A, breast cancer. Option B, colorectal cancer. Option C, cervical cancer. Option D, oral cancer. You have to choose the cancer which is caused by a virus induced, and correct answer for this is option C, that is cervical cancer. Okay, cervical cancer is caused by a virus, and if I have to give one example, that is HPV, human papilloma virus. Okay, this human papilloma virus, it causes cervical cancer, and it is going to transfer from a person to a person during sexual intercourse, and this is the most common cervical cancer. Now moving on to the next question, 26th question. Which of the following destabilizes Hardy-Weinberg law in population? Option A, absence of natural selection. Option B, random mating of individuals in the population. Option C, migration of individuals from one population to another. Option D, absence of mutations in the population. To answer this, first we will know what is Hardy-Weinberg law. I will give the definition now. Hardy-Weinberg law, it states that in a large random mating population, the genotype and allelic frequency remain constant in the absence of any evolutionary influences from one generation to another generation. This is the definition of the Hardy-Weinberg equilibrium. Hardy-Weinberg law should be in equilibrium, means then there shouldn't okay, there is no natural selection, and there is no genetic drift, and there is no sexual selection, and there is no migration. All these will help in the maintaining the equilibrium, or you can say it will help to maintain a constant. Okay, but here in this question they are asking destabilizes. So destabilize ಮಾಡೋದು ಯಾವುದು ಅಂತ ಹೇಳ್ಬಿಟ್ಟು ನಾವು ಚೂಸ್ ಮಾಡಬೇಕಾಗುತ್ತೆ. ಆಪ್ಷನ್ ಎ ಏನ್ ಹೇಳುತ್ತೆ, absence of natural selection. So is absence of natural selection destabilizes the Hardy-Weinberg law? See, if you think, see, Hardy-Weinberg law, think it is in the equilibrium like this. To stay like x-axis and this is y-axis and this represents the equilibrium. If it has to stay like this in equilibrium, then there should be an absence of evolutionary influences. Okay, like there is no natural selection, and there is no migration or a gene flow, and there is no genetic drift, and there is no sexual selection. All these things should be there. Then only Hardy-Weinberg equilibrium will be constant. Here you can say it will be in the equilibrium. If there is any influence of evolutionary influence, then Hardy-Weinberg equilibrium will be destabilizes like this. Okay, Hardy-Weinberg equilibrium will be like this. It will be start to destabilize. What are those they are asking? And in the first option they are given absence of natural selection. Can you guess just now I have given the definition? Is option A is correct? Absence of natural selection. Obviously, absence of natural selection, it makes the Hardy-Weinberg equilibrium manner. Okay, it doesn't destabilizes. Okay, option A correct ide. Okay, the equilibrium na maintain ಮಾಡುತ್ತೆ. Next, second one, random mating of individuals in the population. Yes, if it has to stay in a equilibrium state, then random mating should be there. So this is also correct answer. And it has to maintain equilibrium means there is no migration or there is no gene flow. But here, migration of individual from one population to another anta ಹೇಳ್ತಿದ್ರೆ might be. This is the factor which destabilizes. So option C is the correct answer. And fourth one, what they are saying, absence of mutation in the population makes Hardy-Weinberg law in a equilibrium state. Correct answer for this is option C. If it has to make the equilibrium, then there should be no migration. Here migration of individuals from one population to another. Hagagi this is the one which is going to destabilizing the law. Option C is the correct answer. Moving on to the next question, 27th. Synthesis of DNA strand occurs in option A 3 to 5 dash direction, option B 5 dash to 3 dash direction, option C both the direction, option D either 3 to 5 dash direction or 5 to 3 dash direction depending on energy budget. So this is very easy agide, direct question ide. If you know the concept you can answer directly, and correct answer for this is option B. Okay, the synthesis of DNA strand, it is always going to occur in a 5 dash to 3 dash direction. Moving on to the next question, in a diploid organism the mitotic cell division is characterized. They are asking the mitotic cell division in a diploid organism, how it is going to characterized. Option A, extended anaphase. Option B, absence of chromosome condensation. Option C, formation of haploid daughter cells. Option D, formation of diploid daughter cells. See, they are asking in the mitotic cell division, is there any extended anaphase in the mitotic cell division? No, this answer is not the correct one. Next one, second one, absence of chromosome condensation. So if there is a absence, see, this is also very irrelevant option. Absence of chromosome condensation anta ಹೇಳ್ತಾ ಇದ್ದಾರೆ. That is not the characteristics. B is also more irrelevant. relevant. These two are more relevant. And coming to option C, formation of haploid daughter cell. Haploid daughter cell, and here they are saying diploid organism, and the division is mitotic cell division. Option C is also not a correct answer. The correct answer for this is option D, that is formation of diploid daughter cells. So mitotic cell division ಆಗಿರೋದ್ರಿಂದ there should be a formation of diploid daughter cell. Option D is the correct answer. Now moving on to the 29th question. The molecular formula of immunoglobulin E is option A K2 lambda 2 delta 2, option B K2 or lambda 2 and gamma 2, option C K2 or lambda or epsilon 2, and option D K2 or lambda 2 plus mu 2. And here they are asking immunoglobulin E, so hagagi option C is the correct answer, that is epsilon. It is there in the option C. And let us solve some other as well. Option A is delta. This is the formula of the immunoglobulin IgD. Okay, delta D represents the delta. Okay, and coming to the gamma, this is the immunoglobulin G. Okay, the formula for the immunoglobulin G, and mu2 represents it is the IgM. Okay, immunoglobulin M. Okay, these are the formula of those. And so epsilon, it is immunoglobulin E. Now moving on to the next question. 30th one, facultative heterochromatin is characterized by option A presence of repetitive DNA, option B transcriptional inactive in some cell types in a organism, option C absolute transcriptional inactivity, option D transcriptional activity in all cell types in an organism. Before answering this, first we will know what is this heterochromatin. Heterochromatin, it will be constantly in the inactive state. Okay, and again this heterochromatin is again will be in the 2 state. Okay, and that will be constitutive heterochromatin, and second type is the facultative heterochromatin. Okay, this heterochromatin, I am repeating again, don't get confused. This heterochromatin, it will be in the transcriptional inactive state, and again in this heterochromatin there will be 2 types, that is facultative heterochromatin and constitutive heterochromatin. And this constitutive heterochromatin, it is always inactive, and facultative heterochromatin it will be active in some cells and in some organism, and it will be inactive in some cells and in some organisms. Correct answer ಯಾವುದು ಅಂತ ನೋಡೋಣ ಇವಾಗ. Presence of repetitive DNA, option A, no, it is not the correct answer. Option B, transcriptionally inactive in some cells types in an organism. And coming to the option B, yes, option B is the correct answer here. Transcriptionally inactive in some cell types in an organism. So because it is a facultative heterochromatin. Option C, absolute transcriptional inactivity. No, absolute transcription inactivity ಏನಲ್ಲ. So and coming to the option, transcriptionally active in all cell types in an organism. See, I said facultative heterochromatin means in some cells it is active, in some cells it is not active, it is inactive. And here ಈ ಕ್ವಶನ್ ಬಿ ಆಪ್ಷನ್ ಮತ್ತು ಡಿ ಆಪ್ಷನ್ ಕನ್ಫ್ಯೂಷನ್ ಆಗುತ್ತೆ. ಸೊ ಓದ್ಬೇಕಾದ್ರೆ ಕೇರ್ಫುಲ್ ಆಗಿ ನೀವು ಓದ್ಬೇಕಾಗುತ್ತೆ. ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನಲಿ ಆಕ್ಟಿವಿಟಿ ಇನ್ ಆಲ್ ಸೆಲ್ಸ್ ಅಂತ ಹೇಳಿದ್ದಾರೆ, ಬಟ್ ಐ ಸೆಡ್ facultative heterochromatin will be inactive in some cells and it is active in some other cells. Now I'm not saying all the cells. Okay, option D is not the correct answer. So correct answer is option B. Now moving on to the next one. Who won the Nobel Prize for the discovery of G protein? Option A, Thomas Cech and Philip Sharp. Option B, Rodbell and Gilman. Option C, Carey Mullis. Option D, Arthur Korenberg. And correct answer for this is option B, that is Rodbell and Gilman. He is the one who found out the discovered that the G protein. G proteins are nothing but these are guanine nucleotide binding proteins, and these G proteins acts as a molecular switches. Like these G proteins acts as a molecular switches inside a cell, and these play a crucial role in transferring the signal from various stimuli from outside of the cell and transmitting it to the interior, means to the inside of the cell. Okay, that is the function of the G proteins, and these G proteins will be coupled with GDP, and which is converted into GDP. And this G protein is discovered by a scientist named called as Rodbell and Gilman. That is the correct answer. Moving on to the next question. Bacterial recombination mediated by bacteriophage is called option A, transdetermination, option B, transformation, option C, transcription, option D, transduction. And remember, whenever it comes, it talks about bacteria. Okay, so bacterial recombination, which is going to mediated by a bacteriophage, then it is a transduction. So option D is the correct answer. Moving on to the next question. 33 question. The chromosome abnormality in individuals with Down syndrome belongs to the category dash. Option A trisomy, option B nullisomy, option C gene duplication, option D tetraploidy. The chromosomal abnormality in individuals with Down syndrome it belongs to a trisomy. So here what happens, the 21st chromosome, this one will be added with the extra chromosome. Okay, that actually forms the trisomy. For example, if I have to show this is the Down syndrome, which is a trisomy of 21st, and here you can see the 21st chromosome, which there should be only 2 chromosomes, but one extra one is added here, or it may be partially cutted, or extra one is added. That actually forms the trisomy. It actually causes the Down syndrome in the children, and they looks very abnormal, and there will be a protruded tongue and the puffy eyes. The child will be mentally retarded. So those are the some of the symptoms of the Down syndrome. Moving on to the 34th question, the preponderance of heterozygous individuals in a population is referred as. Option A density dependent selection, option B frequency dependent selection, option C heteroselection, option D kin selection. Kin selection is nothing related to genetics here. Kin selection which comes under the evolution, so option D is not the correct answer. And coming to the density dependent selection, no, this is not the density dependent selection, and it is not at all a frequency dependent selection here. Actually this is option C is correct. This is heteroselection. What is preponderance? Preponderance means it is a which is greater in number. There is a greater in number of heterozygous individuals in a population, means obviously it is a heteroselection. Moving on to the next question. One of the most useful methods for identifying a specific gene is option A Eastern blotting, option B Western blotting, option C Southern blotting, option D Northern blotting. Which one is the very useful method to identify a specific gene anta kelta irodu? First option, first we will solve one by one. Southern blotting, option C is the correct answer. So Southern blotting is the one which is going to help in this identify the specific gene, because it is going to Southern blotting which helps in the specific DNA. Okay, DNA ಇದನ್ನ ಐಡೆಂಟಿಫೈ ಮಾಡಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. ಅಂಡ್ ಅಪೋಸಿಟ್ ಆಫ್ ದಟ್ ಸದರನ್ಗೆ ಅಪೋಸಿಟ್ ಯಾವುದು ನದರನ್ ಅಲ್ವಾ. ಸೊ ನದರನ್ ಇಟ್ ಆಕ್ಚುಲಿ ಸೇಸ್ ಅಬೌಟ್ ದ RNA, ಸ್ಪೆಸಿಫಿಕ್ RNA ಐಡೆಂಟಿಫಿಕೇಶನ್ಗೆ ಅದರಲ್ಲಿ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಅಂಡ್ ದೆನ್ ಕಮಿಂಗ್ ಟು ದಿ ನೆಕ್ಸ್ಟ್ ಒನ್ ಈಸ್ ಸೊ ವೆಸ್ಟರ್ನ್ ಬ್ಲಾಟಿಂಗ್ ಅಂದ್ರೆ ಇಟ್ ಈಸ್ ಐಡೆಂಟಿಫಿಕೇಶನ್ ಆಫ್ ಎ ಸ್ಪೆಸಿಫಿಕ್ ಪ್ರೋಟೀನ್ ಅಂತ ಹೇಳ್ಬೋದು. ಅಂಡ್ ಈಸ್ಟರ್ನ್ ಬ್ಲಾಟಿಂಗ್ ಮೀನ್ಸ್ ಇಟ್ ಈಸ್ ಅ ಪೋಸ್ಟ್ ಟ್ರಾನ್ಸ್ಲೇಷನಲ್ ಮಾಡಿಫಿಕೇಶನ್ ಆಫ್ ದ ಪ್ರೋಟೀನ್. ಅದನ್ನ ಐಡೆಂಟಿಫೈ ಮಾಡೋ ಆ ಮೆಥಡ್ಗೆ ಈಸ್ಟರ್ನ್ ಬ್ಲಾಟಿಂಗ್ ಅಂತ ಹೇಳ್ತೀವಿ. ವಿಚ್ ಆಫ್ ದ ಫಾಲೋಯಿಂಗ್ ವೆಕ್ಟರ್ಸ್ has been most successful for the introduction of DNA into humans? Option A retrovirus, option B yeast plasmid, option C bacterial plasmid, option D tDNA. Correct answer ಬಂದ್ಬಿಟ್ಟು option A for this, that is retrovirus. Okay, this is the one which is most successful for the introduction of DNA into the human cells. Okay, moving on to the next question. Which of the following is not, you're asking which is not the characteristics of normal cells in the culture? So normal cells culture ಇಂದು ಯಾವುದು ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ ಅಲ್ಲ ಅಂತ ಕೇಳ್ತಾ ಇದ್ದಾರೆ. ನಾರ್ಮಲ್ ಸೆಲ್ ಅಲ್ಲ ಅಂತಂದ್ರೆ you have to remember one more scenario. If it is a normal cell, then it should be a cancerous cells. So if you have studied about the characteristics of the cancer cells, so that will be more easy to differentiate. Okay, and also normal cells ಏನೇನು ಕ್ಯಾರೆಕ್ಟರ್ ನ ಪ್ರೊಸೆಸ್ ಮಾಡ್ತವೆ ಅಂದ್ರೂ ನೀವು if you remember one of the characteristics of the normal cells or one of the characteristics of the cancer cells, so anyone ನಿಮ್ಗೆ ಯಾವ್ದಾದ್ರ ಬಗ್ಗೆ ನಾಲೆಜ್ ಇದ್ರೂ ಈ ಕ್ವಶನ್ ಈಸಿಯಾಗಿ ಆನ್ಸರ್ ಮಾಡಬಹುದು. And here they are asking, remember which is not. Option A they don't require substratum, option B they lack contact inhibition, option C they grow rapidly, option D they don't possess aneuploidy. When you look at the options here, see here option A, when you look at the option here, they don't require substratum ಅಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ. Actually normal cells ಏನಾಗುತ್ತೆ, require substratum. Okay, and here they are also not asking which is not. Wrong answer ಅವ್ರು ಕೇಳ್ತಾ ಇರೋದು, and they are also written here they don't. So ಹಾಗಾಗಿ option A is the correct answer. Next option B, they lack contact inhibition. Normal cells ಏನಾಗುತ್ತೆ, they have a contact inhibition so that they don't produce more excess or something like that. Here they are saying they lack, ಹಾಗಾಗಿ even option B is also correct. You can option A or option B, so because both the answers are correct here. Option C they grow rapidly and they don't possess aneuploidy. These two are not correct. Moving on to the next question, 38 one. Direct cytoplasmic communication between neighboring cells is facilitated by option A gap junction, option B tight junctions, option C desmosomes, option D Golgi apparatus. So Golgi apparatus ಅಂತೂ ಇದ್ರಲ್ಲಿ ಬರೋದೇ ಇಲ್ಲ, ಸೊ ಹಾಗಾಗಿ option D ಅಲ್ಲ. And coming to the ಇಲ್ಲಿ ಕ್ವಶನ್ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳೋಣ. ಡೈರೆಕ್ಟ್ cytoplasmic communication between neighboring cells ಅಂತ ಹೇಳ್ತಾರೆ. Think this is one cell and this is the another cell. This direct communication between these two neighboring cells is facilitated by actually a gap junction. Okay, option A is the correct answer. Moving on to the next question. Human sperm nucleus contains option A 22 pairs of autosomes plus X or Y chromosome, option B 22 autosomes plus X or Y chromosome, Option C, 22 autosomes plus X and Y chromosome. Option D, 22 pairs of autosomes plus X and Y chromosome. Here they are saying human sperm nucleus. The correct answer easy agide. You have to be observing keenly here. Option B is the correct answer. 22 autosomes plus X or Y chromosome. Okay, that is the correct answer. Moving on to the next question. 40th one, standard error is calculated by equations. E optionally, E4 optionally, option D is the correct answer. This is the equation of the standard deviation, SD divided by square root n. Okay, this is the equation for the standard error. And I'm hoping this video is going to help you all. If you learn something from this video, please do share and subscribe. In the next video, I'm going to solve the rest of the questions. Thanks for watching.